Hello, everyone. Welcome to Nature and Science Podcast, where science does care what you believe. I'm your host, Don Murphy. We explore the science behind the latest technological developments, ask the deep questions about existence, and seek verifiable truth leading to a clearer view of reality. On this episode, we explore one of the most enduring mysteries in cell biology, the nanovault, a small protein capsule within the cell. No one seems to know why they exist or what they do within the cell. Even cell biology textbooks have ignored their existence. Dr. Leonard Rome and his colleague Dr. Nancy Kurdersha discovered them in 1986. Dr. Rome has been working to solve this mystery ever since. He joins us to share his journey and long relationship with vaults. He's the distinguished professor at the Department of Biological Chemistry at the David Geffen School of Medicine at UCLA, Associate Director at the California NanoSystems Institute. He's the Chief Scientific Officer at Vault Pharma. Rome earned his BS in chemistry and MS and Ph.D. in biological chemistry at the University of Michigan, Ann Arbor. Hello, Dr. Rome. Welcome to the program. It's great to have you with us today.
Great to be here, Don. Thank you.
So I'm always interested when I talk to scientists and finding out what started them on their journey as a scientist. Was there something that happened to you as a youth or something in your past that said, hey, this is what I want to do?
I don't think it happened that way. When I grew up in Youngstown, Ohio, and I was just a incredibly curious kid. I had every hobby imaginable. I collected stamps. I did a lot of artwork. My mom was had a lot of talent in that regard. I just did every hobby, and so when I went off to college, I had a very broad interest in doing everything.
That's great. And so somewhere along the line, you said, biology and biochemistry is it.
Well, science. I certainly tended toward having a broad interest in science. One of my first summer jobs when I got into college was working in a small laboratory in Youngstown for a doctor who was trying to do research. He wasn't a trained researcher, but he was interested in a new field called prostaglandins. And I worked in his lab, and I was just taken by the process of doing research, and that kind of got me stimulated about research.
So this program is all about vaults, and I got so excited when I read about vaults and some of the information of your background, because I love a scientific mystery. I mean, I started my career out as a researcher at the Salk Institute, and mysteries have always fascinated me in science. And seems like one of the greatest mysteries in science. And of course, there are many, but this is pretty intriguing. So we're here to talk about vaults. So why don't you start off by telling us what a vault is and how you got involved in discovering these little protein packets that have started you on a 30-year quest to understand them.
Yeah, that's a big question, but I'll certainly try my best. I wasn't looking for vaults. Okay, I'm a trained biochemist in terms of my Ph.D., but when I did my postdoctoral research, it was at the NIH in a laboratory that studied inborn errors of metabolism, especially lysosomal diseases. And these are a whole group of terrible diseases in kids where they have a problem breaking down different complex carbohydrates. And so I got trained as a cell biologist during my postdoctoral years at the NIH in a laboratory of Elizabeth Neufeld. And so when I set out to start my own lab and got hired at UCLA as an assistant professor, I continued to work on lysosomes and trying to understand this organelle. And my interest was in trying to figure out how enzymes got transported through the cell into the lysosome. And in order to do that, we had some hypotheses at the time that these enzymes might be being transported in these small round transport vesicles called coated vesicles. And so my lab was purifying coated vesicles, looking for lysosomal enzymes that might be transported to the lysosome. And in order to do that, we were fractionating coated vesicles into more distinct fractions in order to study this.
Sure.
And we just stumbled upon a particle that looked a little like a coated vesicle. It wasn't round, though. It was sort of oblong, football shaped. And we thought it was related to coated vesicles. So we started to study it. And it took us about 6 months or so before we realized that this particle had nothing to do with coated vesicles. It was completely different. But by that time, we were hooked. We just wanted to know more about it. I mean, there was really no aha discovery moment. It was like we found this thing. We thought it was related to coated vesicles. We started studying it, looking at its structure, and eventually we realized we had found something new. And that led to a whole bunch of questions. How common was it? How abundant is it in cells? What should we call it? All those questions came up.
So why did you call it vaults?
Yeah, so this is one of the most difficult things to describe on a podcast because obviously, if we had a picture of the particle, we could see it. But actually, the three-dimensional structure of the particle itself wasn't something we had when we found the particle. What we had were some images in the electron microscope of this particle. And the images of the particle in the electron microscope were carried out using a technique called negative staining. And this technique is when you're looking at a particle that doesn't really attract stain itself. The vault particle is mostly a protein particle, although it has a small RNA component. It's mostly protein. And so under an electron microscope, it's kind of transparent. It doesn't really diffract electrons. So in order to see it, we pour a dense stain over the particle. It's a heavy metal, uranyl acetate, and this stain forms puddles around the particle. Dark areas of stain aggregate around the particle, and that's called negative staining. And when we negative stained this particle, it appeared to have arches that reminded us of the vaulted ceilings of cathedrals.
Oh, really?
And that's what led to the name vault. We actually had a contest in the lab, Name That Particle contest. Right. And many people in my lab wanted to name the particle a romosome, and I didn't think that was a good idea. It's not usually, naming something after yourself doesn't usually stick, okay? I don't know if you know this, but there are several particles that people tried to name after themselves, and that didn't stick. And so I like the idea of naming it more like its morphology. And since it looked like these vaulted ceilings of cathedrals, vaults is the name that we stuck with.
Well, at the end of the podcast, I'll have a link both to your YouTube channel and to my website, and people will be able to go and see what these vaults look like. Right. So even though we're talking about them here on the podcast, right. And then I'll also be publishing this as a YouTube podcast, and there will be some photos there as well that people will be able to see. So that'll be good because they're absolutely fascinating structures. So what lines of investigation have you done to find out what these vaults do? So I would imagine the first question that came to your mind is, what are these things there for?
That's right. It was one of the early questions, but first we asked the question, how common were they? And that was the first surprise, that every cell that we had in the laboratory, we started thawing out cells from our freezer and looking at, we did a lot of cell biology, so we had a lot of different cell lines in the freezer. But I mostly worked on eukaryotic biology, and it turned out that vaults are highly expressed in eukaryotic cells. They're
Mhm.
not in prokaryotes.
Yeah, just so people know that are listening, I always try to make sure they understand when you're saying eukaryotic cells, you're talking about cells that have a nucleus, and
Yes,
prokaryotic cells like bacteria, like E. coli, don't have a nucleus, and that's the difference. So you're end up being focused on eukaryotic cells, cells that have a nucleus.
that's right. And so we looked in tissues. So we were working in on mouse and rat models in the lab. So we looked at the tissues of the mouse, and we found that vaults were in every tissue of the animal. And when we looked in the cells that we cultured in the lab, vaults were in the cytoplasm of the cells, not in the nucleus, and not they didn't seem to be aggregated in any particular organelle. They were in the cytoplasmic
Mhm.
soup of the cell. And the other surprise was there were a lot of them. We calculated about 10,000 volts in the average cell,
My
in the
goodness.
cytoplasm of the average cell, which made volts one of the most abundant particles of the cell. And so that was the first big mystery. How is it that no one had described one of the most abundant particles of the cell? And that was also true of human cells.
Yeah. But that leads me to ask, and this was a question I didn't ask you ahead of time, but if or maybe it's coming up, is if you have that much mass
Right.
uh, or bulk, if you will, for my listeners within a
Yep.
cell, it leads you to think that the vaults, just their mere presence in numbers, have something to do, maybe with the morphology or the structure of the cell itself. It's somehow holding the cell together. So I'm sure in your lines of investigation, you're far more sophisticated than I am. You must have asked that
Well,
question.
we have asked that question, and it turns out that the vault particle itself, even though it's a huge particle, it's on the order of 3 times the size of a ribosome. And so most cell biologists obviously know what ribosomes look like because it's one of the few particles in a cell that does stain in the electron microscope. And the reason that you can see these little black dots in the cytoplasm of cells is that the ribosome has so much RNA associated with it that when the cells are stained, usually by a cell stain like osmium, the RNA stains very darkly. And so ribosomes appear as these little black dots. Although vaults may be 3 times bigger than a ribosome, they don't stain. So you just
Hmm.
don't see them. They're kind of
Hmm.
in the background. They're kind of like gray blobs in the
Right.
background of the cell. So, as far as the mass of the vaults and the amount of vault protein probably makes up less than 1% of the total protein of a cell. So they're not really that much of an occupant, although pretty significant, not that much. And
Mhm.
later, when we were able to knock out vaults in cells, they didn't really affect the phenotype of the cell. So vaults are not doing some essential function that can't be replaced by other proteins of the cell.
Right. Well, when you say essential function, I know this question was going to come up later, but now it's coming to my mind now, and I hope you
Sure.
don't mind. It's
No, I don't
something that
mind.
we talked about earlier. If it's not if it apparently is not performing an essential function, perhaps it's performing a function under certain conditions,
Yes.
abnormal conditions. So it led me to ask you, of course, whether or not under some abnormal condition in the cell that you don't normally see and that may be very rare, the vault may have some function.
Yeah, that's the big mystery, and so far we haven't figured out what that condition is. It reminds me of a story once I heard of a protein knockout in a mouse that had been carried out by a group, and there was no phenotype. And then one of the uh, people in the lab that was handling a cage of the mice accidentally dropped a cage just about a foot down onto the bench, an activity that wouldn't bother a normal mouse, and the knockout mouse died. And so they got this protein had something to do with the ability of that mouse to absorb a little shock. So unfortunately, we haven't found the condition that tells us what the vault might be essential for. But I agree with you. With so many vault particles in every cell, there must be some reason why they're there. People have challenged me. They say, well, the vaults might just be the appendix of the cell and it's no longer needed. And that's possible, but evolution argues against that for a number, and I can raise a number of points. There are some cell types that have a lot of vaults where it's an evolutionary advantage for them to get rid of genes that they don't need. So a long time ago, we studied vaults in an organism I'm sure you've run into before, Dawn, and that's the slime mold, Dictyostelium discoideum. And we were studying vaults in Dictyostelium because it's an interesting developmental model. You can put this slime mold through its developmental sequence of events, and you can look at proteins and ask, are they going up and down? And that might give you some hint as to what their function is. And so we actually cloned the first vault protein, the major protein of the vault particle, which I haven't got into yet, but I'll come back to it. We started by cloning this protein and looking for its expression during Dictyostelium development. And what we found was it was present all through Dictyostelium development, even in the spore. And in Dictyostelium spores, Dictyostelium has made its genome very small so that its spore is very light, so it can fly through the air. And so why it would preserve the vault particle even into the spore suggested that it was important to the Dictyostelium at least. So getting back to the vault particle, even though it's a massive particle in 3 times bigger than a ribosome, 13 million Daltons in molecular weight, it's dominated by a protein of about 100 kilodaltons that makes up 70% of the mass of the particle. And it's the major structural protein of the particle, and it makes up the entire shell of the particle. And we call this protein the major vault protein, or MVP for short. And we knocked out the MVP protein in Dictyostelium, and we found out that it had 2 genes encoding the major vault protein. So we had to knock out 2 major vault proteins in Dictyostelium to get rid of the vaults. And the Dictyostelium was fine, except it did grow more slowly than normal Dictyostelium. And that led us to suggest that maybe vaults had something to do with the growth of the Dictyostelium. And it has led to some people speculating that maybe vaults are a storage form for amino acids. And in Dictyostelium, maybe they need vaults to store amino acids during their life cycle. And that may be the function of vaults in Dictyostelium. But obviously, vaults have been carried on throughout evolution into higher eukaryotes like man, and they're more complicated. They have 2 other proteins inside the vault particle, 2 very high molecular weight proteins. One is called TEP1, which has been found in the telomerase complex, which is crazy. It has nothing to do with a cytoplasmic particle. And the other is an enzyme called VPARP, or VaultPARP, which is a member of a family of enzymes that carry out a protein modification reaction called ADP ribosylation. And so the finding of these 2 high molecular weight proteins inside the vault and a small untranslated RNA called vault RNA inside the vault also leads me to think that vaults have some other functions, higher functions in eukaryotes.
Yeah, I mean, the fact that it has this small snippet of RNA is just absolutely fascinating. But I suppose others, not necessarily critics, but those that, would challenge the notion that there's still some function for vaults, particularly since evolutionarily they seem preserved. And you were saying there were two genes involved in the production of one of these proteins, and that the slime mold, its growth was slowed down. I mean, others could argue, of course, that some genes have a multiple function, if
Yeah.
I'm, not mistaken, and so those very genes could be involved in some other function in the cell, other than just the production of the protein for the vaults.
Yeah, I'm glad you brought that up because obviously, when you find a new protein or a new particle, the first thing you do is you ask, well, what is it similar to? You find a new protein, you say it's a member of a
Right,
protein family. Is it
right,
an enzyme? Is it a kinase? You get some clue from the protein itself. And so again, another one of our major setbacks was when we finally did clone the gene for the major vault protein, this major structural protein of the vault that makes up 70% of the vault mass. It was related to no protein in the database. There is
huh,
no family of proteins that the major vault protein is similar to. Now, of course, as genomes were sequenced throughout, we found vaults back in the 80s. And so during that time, there weren't any genome sequences published. But in the 90s, people were publishing genome sequences all the time. And we started to compare the major vault protein to different genomes. And we found that they were only related to major vault proteins in other genomes and not
hmm,
to any other proteins in other genomes either. And
right,
this really was a puzzle to us as well. And one of the things that solved this puzzle, well, didn't solve it, but added to it, was when we figured out how vaults were made in cells. And this is another, I think, amazing story. And that is that the major vault protein is not made as a free protein in cells. So if you think about similar structures, viruses, viral capsids come to mind when people say, well, what things look like vaults? Well, vaults are a protein capsid, and so made up of a single protein in multiple copies to make up the outer shell of the vault. In fact, 78 copies of the major vault protein are assembled into the vault capsid, vault's
right,
shell. And so
sure,
you ask, well, viruses make protein capsids, and how do they make them? Well, they produce a lot of that capsid protein, and the protein self-assembles into the capsid. And we thought the vault particle must self-assemble from multiple copies of the major vault protein into the vault shell. And that's not how it works at all. When we looked for free protein, free major vault protein in a cell, we couldn't find any. And in fact, the reason is that we don't think there is any major vault protein free in a cell. The vault is made one vault at a time on a structure in the cell called a polyribosome, a
sure,
well-known structure that is involved in protein synthesis. But people usually think of a polyribosome as a structure that makes proteins one at a time. It
sure,
chugs them out one at a time. But what happens is that the major vault protein is made on the polyribosome, but it stays attached to the polyribosome until 78 of them are formed. And it actually forms the vault structure while attached to the polyribosome. And so the cell makes vaults one vault at a time from on a polyribosome.
right. That's really fascinating. You have a visual on your YouTube channel of how this is done, and I mean, I give the kudos to the animator. You should have won an Academy Award. It's just so I'll have a reference so that people can actually look and see this how these structures are made, because it's really just absolutely fascinating, and it's in color, so you'll get to see a color
Yeah. Yeah.
representation of how these vaults are made, because it's hard just articulating what it looks like, but when you see it, it's just absolutely fascinating.
Yeah, you are so right. I started to describe this process to my colleagues, once we thought we had figured it out, I would give seminars and I would tell them about how the polyribosome is making a vault. Nobody got it. And then
Right.
finally I had no funds, so I scraped together enough of my research money to hire a guy who had never animated a biological a thing before in his life, and I worked with him to make that animation. And then as soon as we made the animation, everyone I would show it to immediately got it. And
Right.
so you're so right. It made all the difference in the world.
Yeah, I was glad before I talked to you that I got a chance to see that animation because I was just glued to the screen because it
Right.
was just so fascinating to watch this sort of thing. And those of us that have worked in cell biology, for years or microbiology, when we see something like this that's totally out of the realm of what we've ever, seen before, you're just, aghast at how fascinating it happens to be. So I asked you a kind of a joking question earlier, with the smiley face, and I said, so perhaps the cell is somehow paying homage to these vaults. They had some sort of function in the past, but now they're kind of obsolete, and they're being kept around because they were so important that, and I kind of compare them to the monarchy. The monarchy is
Yeah.
in many countries is obsolete. We keep them around because they, somehow remind us of maybe a more glorious past or something. And so I was wondering if the cells just said, "Hey, we like you and we're going to keep you around."
Well, it does require a fair amount of energy to make a vault, assemble this huge particle, and then to keep it around. The half-life of a vault is quite long, weeks, maybe even months in some cells. And so, even though vaults are made slowly off of maybe on average one messenger RNA in the cell, they tend to accumulate into this tune of 10 to 20,000. And in fact, some cells have 100,000 volts.
Wow.
These would be immune cells. So like a macrophage has 100,000 volts. So it would seem to me pretty inefficient for the cell to pay homage to the vault by keeping around so many so many vault particles. It might be a little bit more efficient to lose that gene if it could, and it doesn't seem to want to do that.
Well, except that some anti-monarchists make the same argument.
I can't, Don, I cannot count out the fact that that might be what's going on here. We have been studying mice that have had all of their vault proteins knocked out,
Hmm.
and uh, these mice appear to be normal, but they have some subtle problems, and mostly those problems are with their immune system. So it is leaning us toward thinking that vaults might have a fine tuning kind of function in the immune system.
Right.
And I think another analogy to bring up would be that if you had a really sophisticated amplifier that you were playing music from, audiophiles love to have a lot of knobs on their amplifiers.
Right.
And so they tend to have a coarse volume control and a fine volume control, right?
Mm.
So you can get the volume right with the coarse volume control. It's just a lot more work, right?
Right.
So I think of vaults maybe as one of the fine volume control knobs. And if we get rid of that, maybe the amplifier still works. It just doesn't work as well as it should.
Right. Well, I'm going to go out on a limb and support you on that and say that I think that that function will probably be found at some point. And those folks, I mean, I had colleagues over the year and still do who work on the immune system, and it's to say it's complex is an understatement.
Yes.
So it's more than complex, and so it certainly would make sense that might be the case. Well, let's switch reels. And so, since you have not figured out what they seem to do in the cell, you have been working on some adaptive use
Yes.
for these vaults, perhaps in drug delivery and that kind of thing. Why don't you talk about that for a minute and tell us what you've been working on in that regard with vaults?
Yeah, this was sort of invention. Necessity is the mother of invention, I think is the
Right.
saying. And so we had been working on vault function for 15 years when the funding sources kind of dried up for us. I think the NIH lost interest in vaults when we couldn't figure out what they were doing. And so I was searching around trying to keep my vault research funded. And I had this thought that maybe instead of figuring out the function of vaults, maybe we would give them some functions. And
Right.
so that led to a whole new direction for my lab, which was to try to figure out how to modify the vault particles to give them some new uses. And
Right.
so that was enabled by the finding that we could make empty vaults, that we could make vaults that were just composed of the shell of the vault, this major vault protein. And so now we had a vault that was 100% made of the major vault protein because it didn't have any other vault proteins inside. And we did this originally in a cell that didn't have vaults. So even though I told you that vaults were highly expressed in eukaryotic evolution, it turns out that there are some very notable eukaryotes that have lost vaults and
Hmm.
don't have them at all. So
Oh,
an entire kingdom, plants, have no vaults. Uh, but also there are some animal eukaryotes that don't have vaults. They aren't found in yeast. They aren't found in worms like the common worms that are studied in the lab. They aren't
like
found.
nematodes.
Yeah, and they aren't found in most insects, although they are in some insects.
Hmm.
So we were studying a protein expression system that's done in insect cells. Biologists use it all the time. It's called the baculovirus expression system, and it uses an insect cell to make proteins. And that's how we discovered that if we expressed the human major vault protein in an insect cell, it didn't it didn't make the protein, but it made vaults. It made empty human vaults in the insect cell. And
Hmm.
that led us to understand that we could make empty vaults. And we now make empty vaults in yeast because we also learned how to take common yeast and
Right.
express the human major vault protein in yeast and get empty vault particles. But the key to giving the vault new functions was figuring out how to package a protein inside the vault.
Right.
And one of my close colleagues and started out as a postdoc in my lab, Valerie Kitkofer, who worked with me for 30 years on vaults, she figured out how one of the vault proteins got inside the vault, this vault protein called VPARP, and it had a domain on the protein near the C terminus of the protein that we called the major vault protein interaction domain, or INT for short. And she figured out that she could take this domain, INT, and attach it to other proteins, make fusion proteins. And
Hmm.
those other proteins were now directed to the inside of the vault. This
Oh,
domain acts like a zip code. And if you attach it to any protein, it directs that protein to the inside of the vault particle. And this was a
hmm.
great advancement for us because the first thing we did was we attached this zip code to an enzyme, luciferase, the enzyme that gives the
Sure.
firefly its glow.
Right.
And we succeeded in making little nano light bulbs because now the luciferase enzyme was packaged inside the vault, and we could add the substrates for that enzyme, and the vaults glowed. Not a very practical light bulb to make, but it did allow us to follow those vaults around in cells because
Right.
they glowed. So using that same technology, we've gone off into different areas for vault applications. Um, One of them has been for drug delivery. And so
Right.
we've attached that zip code to some important immunological molecules called chemokines. These are signaling molecules for the immune system. And one of those chemokines we've put inside a vault, and we're trying to use it to wake up the immune system to identify cancer. And that's the one vault drug that is furthest along in our quest to commercialize a vault particle and make it into a drug. We also have a whole field of research on environmental remediation using
Oh.
vaults.
Hmm.
Yeah, I've been collaborating with a very good group at UCLA, headed up by an environmental engineer named Shally Mendra. And Shally wants to put enzymes into the vault particle that will degrade water contaminants,
Right.
like dyes and PFAS molecules, bisphenol A, for example, these dyes that and chemicals that leach out of plastic. And so Shally and I collaborated to produce vaults containing some enzymes that will degrade these environmental contaminants. And we think that might be a direction that vaults might be useful for as well.
Right. Understand. What about production of what you would call other biomolecules? I know that some of my colleagues are working with bacteria or yeast to produce other biomolecules that would be synthetic oil, for example,
Yeah.
that could be used for fuel in some instances. Is that something you've pursued as well?
We have not, but now that we have the yeast system up and running, it's quite a robust system. The reason why we turned from insect cells to yeast was just so that we could get quantities of material made that would be relevant to, say, environmental engineering. And so with the yeast system running, we can make kilograms of vaults. And so using them for other types of applications is not so far-fetched.
Right.
And so those things maybe should be considered.
No, I understand. So I know we've kind of touched on this question already, but if faults have no apparent function within the cell or the organism of a whole, how does the cell interact with the vaults? Is there any interaction that
Yeah,
the cell has with the vaults, even though it may not be something that helps the cell? There must be some sort of interaction that goes
yeah.
on.
That's a great question, Don, and that's what keeps me up at night. Okay, so the vault particle has all kinds of interactions in the cell. It's just that as fine tuning goes, they're kind of subtle and hard to pin down. Over the past 20 years, there have been many reports in the literature of other proteins that bind to or interact with the major vault protein.
Mm,
Okay, so
mm,
and there's been a number of these molecules that are important. They're important signaling molecules in a cell that people have published data showing that there's an interaction between their favorite protein and the major vault protein. And I think that in many cases, these other investigators have missed the point by not really reading a lot about vaults and trying to understand what the major vault protein is. And so when someone nowadays publishes a finding that says, oh, my protein X binds to the major vault protein, they missed the boat because I told you a few minutes ago, there is no major vault protein in this cell, only vaults. And the real take home message should be my protein X binds to
first of
vaults.
all, right.
Yeah, and so I think that's going to open up a whole new areas of research. And recently, there have been a couple papers published where they realized that it wasn't an interaction with the major vault protein they were seeing, it was an interaction with vaults. And one paper, a recent paper, came out showing that a important kinase was interacting with vaults
Mhm.
inside cells, and that might be a hint that we can follow up. Another came out showing that a cell kinase involved in cell signaling through an integrin was interacting with a vault on the cytoplasmic side. And that too was a mystery that we had seen many years ago. And that is that one of the locations in the cytoplasm where vaults were gathering was at the extreme edges of the cell, what's called the leading lamellipodia in a motile cell. And that's a very thin layer of the membrane with not much cytoplasm, but lots of vaults. And that might be because vaults are interacting with proteins involved in signaling at the cell membrane. So these are really interesting clues, and I hope will lead some of these other scientists to asking questions about, is that interaction that they're seeing due to the function of the vault particle?
So I have to ask, were you excited to point out to them, hey, it's not just interaction with the vault protein, it's actually with the vault. And what was their reaction? Was it they like, oh, that's good. Maybe that's another line of investigation I should follow.
It was. And these two recent papers, I immediately, sent them emails. And you don't know me, but you might be interested in the fact that what you've published is interesting with regard to a particle called a vault. And both of those groups now, I've sent them antibodies, and they're going to be looking more closely at whether or not their signaling cascades are being affected by the vault particle itself. And so, yeah, I'm encouraged that this may be leading to new collaborations. I have a very small lab now. I'm a recalled emeritus professor. I don't write research grants anymore, so I have to depend on collaborations to try to keep moving the vault story forward. So I honestly, I really appreciate the fact that you've discovered vaults, and that'll help me get the vault message out there.
Right. So you mentioned the half-life of vaults. So there must be some sort of disposal system for vaults. And I have a related question as well. But what is the disposal system like? I mean, if the proteins from vaults get broken up, do they get recycled? Is there some periodicity to the death of a vault that might tell us something? What happens there?
Yeah, I'm afraid to say there's very little knowledge about how vault turnover actually occurs, because if you try to study the turnover in a cultured cell, in cell culture, there's just no turnover. Because the vaults are made so slowly, and because they're so stable inside the cell, they're little rocks in the cytoplasm, okay? That when a cell divides, it just splits up the rocks, and half go to the daughter cells and each cell. And so there's little data on whether they're recycled. Well, we know that they can be degraded because there is some turnover, but we don't know if that's in a lysosome or through proteasomes. We just don't know how that works. There may be some ubiquitination, a marking of a vault for degradation in a proteasome. It has not been studied. I think it's an interesting issue, but so far it hasn't been investigated.
So what do you believe is the most or the best working hypothesis that explains the existence of vaults in your mind? I mean, you've been doing this for 30, 40 years.
Yeah, I was afraid you were going to ask me that. Well, I have a couple of pet theories, okay, for vaults. One of them is back to this role in the immune system. Okay, and it requires a little bit of background for me to give you this pet hypothesis. But one of the applications we were using vaults for was as vaccines. So we were putting foreign proteins into vaults, and we were asking, could vaults work like vaccines? And so we were immunizing a mouse with a vault containing a foreign protein, foreign for the mouse. And what we found was that the mouse would raise antibodies against the foreign protein, but it also stimulated a cellular immune response. That is the formation of T cells that were directed against the foreign antigen. And that's not something that usually happens when you inject a protein as an antigen. And so vaults were acting like adjuvants. They were actually waking up the immune system to make a cellular immune response. And vaults themselves are non-immunogenic. So if you uh, 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 immunize a mouse with a human vault, even, the mouse doesn't make antibodies against the vault. Human vaults and mouse vaults are so closely related genetically that there isn't a immune reaction against the vault. But once you put a foreign antigen in the vault, now you get a terrific immune response. And so that fact, and one other tidbit that I want to tell you, and that is that we asked the question, what cells interact with free vaults? And we did this by adding vaults to blood, right? So we just mixed human empty vault particles with human blood, right? And then we used cell sorting to ask what cell in the blood will interact with a vault. And it was very easy. There were only 3 cells in the total blood of all the blood cells that reacted with vaults, and those were phagocytic cells. Blood macrophages, which are called dendritic cells or antigen-presenting cells, activated T cells, and a small subset of B cells that's also phagocytic. So those were the only cells, but they gobbled up the vaults as if they were the tastiest morsel they had ever seen. Within a half an hour, every one of those cell types had taken up the vaults. We got 100% labeling of those cells. A remarkable thing, okay?
But it still wasn't immunogenic.
No,
they were just gobbling them up.
They were just gobbling them up, okay? So that has led to one pet hypothesis, and that is that, normally cytoplasmic particles don't really get into the blood, right? Cells don't burst, okay?
Right.
That is not a normal way for a cell to die, all right? In order for a cell to burst, it's usually because of some trauma, okay? So a wound will open up a cell and cause them to burst, but a pathogen will lyse a cell, okay? And
like
so
a virus.
Like a virus or a bacterial pathogen. And so one pet theory that I have is that when a pathogen lyses a cell, the vaults come out of the cell cytoplasm, and they are gobbled up by immune cells, and they're sending a signal. Okay, and I don't know how they get that signal yet, but we're working on it. But maybe they're sending a signal to the immune system saying, hey, pay attention to me. I just came from a cell that was lysed. And that's why every cell has vaults. They might be a primary signaling system to the immune system that they've been released from a cell that shouldn't be releasing them. So,
well, that's fascinating because it does open up some lines of study and some ways of trying to falsify that hypothesis. So it's possible to do that. I'm going to pay attention and see if there are papers that come out that end up talking about that. I mean, that's just really fascinating. So another difficult question, which may lead you to ask me a question, which I hope I can answer, is could something be going on at the quantum level that's involved with vaults? And I'm thinking about superposition, and I'm thinking about all the complexity, for example, of photosynthesis and how electrons transport happens. And we know that, there's electrons almost make choices. This is the path I'm going to follow. And scientists and physicists don't exactly know how that happens. It's a quantum phenomenon. And so I'm just wondering if there's something happening on the quantum level that vaults are involved with, and if you've asked that question.
Yeah. I've not been able to experimentally ask that question because I don't know the first thing about asking that question. I have read what some of the things you're alluding to about how photosynthesis might involve quantum tunneling, and I think that's really cool. And we have some quantum physicists in our at UCLA that work on these problems. And I just can't figure out a way to ask them how we could look at a vault particle in that regard. Maybe you have some ideas that we could use them for. And I agree with you that the symmetry of the vault particle is what would drive one to ask this question because they have this incredible symmetry. The 78 proteins that align themselves, 39 on each half volt, facing in the same direction, interacting with each other, all the way up this 100 kilodalton chain of protein. It is a remarkable structure. How I could ever figure out how to ask a quantum question about it is something that currently is beyond my imagination.
Well, I'm going to keep thinking about it, and maybe we can have some discussion in the future about it. And it leads me to ask you. I know you're emeritus now. I mean, I'm fascinated by this. I can't imagine how you must feel, not writing grants anymore, not being directly involved in the lab. So, what's the future hold for you? I mean, do you feel imprisoned that you can't break out and be a 30-year-old again? Going gung-ho to find all this stuff out, answer these questions.
Parts of me would like to be, obviously I'd like to be in my 30s again. I have some ideas. I would like to write up some grants, but I think that my role at the moment is to try to get other people excited about vaults. And so you mentioned finding out about vaults. I didn't ask you whether you found out about vaults through my YouTube channel or not, but this is the way that I'm trying to get people interested. It frustrated me that vaults aren't in textbooks. I understand why. There's a lot of textbooks are limited in their number of words you can put into a book, and there's a lot of mysteries in science that we don't know about. And so vaults got left on the cutting room floor when those textbooks were made. And so I think that one of the things that turns me on is just trying to get other people to realize that vaults are out there and vaults are cool. And I think that if they would have showed up in textbooks, there might have been a lot more people out there asking the questions you're asking today. What are vaults doing? What can we do with vaults? Why are there so many of them in all of higher eukaryotes?
Right.
Yeah.
Well, I know I would have been fascinated. Of course, I was in my microbiology classes, long before you discovered vaults. I'm probably 10 or so years older than you. And I in my microbiology class, if I would have seen a vault in a cell, I had a lab partner and we just loved our microbiology class. And this is the kind of thing where we would have said, man, I wonder what that is. And we would have been very excited about that, like you're saying. So it's too bad they're not in textbooks, but maybe that's something that'll change. And there wasn't I think it was a writer of a science magazine article,
Yeah.
and that's where I learned about vaults when I was reading an article in Science.
Yeah, if you have time, I can tell you a quick story about that.
Oh yeah, absolutely.
So back in 1995, there was a young science editor working for a little science magazine called Science News. It wasn't a very big magazine, but
Yeah, I
he
remember.
somehow stumbled upon the vault story and called me up in 1995. His name is John Travis, and he wanted to learn about vaults, and he ended up publishing a nice review in 1995 of vault particles. At that time, they were thought to be involved in cancer cells in a phenomenon called multi-drug resistance. And so John published this really neat review paper about vaults and had a picture of a cancer cell with vault staining on the cover of Science News. So flash forward 30 years, about a year and a half ago, I get a phone call, I get an email from John Travis, and he says, maybe you don't remember me, but I wrote an article about vaults 30 years ago. Now, I laughed to myself when I saw that email because I have that picture framed on my wall. Okay, so I certainly remembered him. But he had seen a preprint from a collaboration that I was involved in where we were packaging a virus particle inside a vault. And so he wanted to talk to me about that. And so we had a Zoom call, and we talked about it. He decided he would wait till that paper was published, but he asked me, well, what's going on with vaults? And so we started talking, and that led him to write yet another article. Now he is the chief editor for Science Magazine. So he has certainly come up in this world. And he wrote what to me was the nicest article that anyone has ever written about me. I felt very fortunate that he wrote an article about vaults and about me in Science.
Yeah, it was a very, very well written article, and I was just fascinated by it. And I said, I'd love to talk to Dr. Rome about this. And I was so gratified that you responded so quickly, because it's the kind of thing that my listeners love and that I like to talk about in terms of science, because this is how basic science is done. And most people are looking at science now and saying, well, maybe that research isn't that important. Well, what are those guys doing off in a lab just working on something? I mean, what's going to come of that? And of course, you never know what's going to come of it. I mean, that's how CRISPR was discovered. Who would have thought? And so this is what basic research is really all about. And it's kind of in that context that I want to present that to my listeners and to others as well.
Yep.
This has been a fascinating conversation, and I really hope to stay in touch with you and find out more about vaults. I'm going to put a little note in my Google search so that whenever something on vaults comes up, I'll get alerted, and I'll be able to see what's going on. And again, in my post talk about this on the podcast, I'll certainly mention your website. I think you're known as the Vault Guy on your on your YouTube site, and so I'll certainly mention that, because a lot of the information is just basic good cell biology information as well, and so it's something that people can really learn from. I really commend you. You do such a great job with that.
Well, thank you. That was the idea. I thought, well, how, I could never explain what a vault is to a non-scientist without them learning a little cell biology, right? So my YouTube channel is just a way to teach you some cell biology, a spoonful at a time, so that you can understand how cool vaults are. So that's really the object of what I'm doing. It's not easy though, and you probably understand this very well. Most YouTube channels are one-off, right? You can just pick one and watch it and get, something out of it, right? I'm trying to tell a story by teaching a sequential series of courses, and that takes quite some dedication for people to watch one episode after the next and to retain the lesson from the previous episode. That's a challenge.
I'm going to do all I can to get the word out. Believe it or not, there are several vehicles that you can use at my YouTube channel, or that my podcast is attached to, that I can get the word out, and I can direct people to your YouTube channel.
I thank you for doing that. I thank you very much.
Not a problem. Dr. Rome, thank you so much for joining us on Nature and Science podcast. And hopefully in the next few years, I'll have you back and you'll be telling me exactly what a vault is and does.
I hope so too, Don. I really do. Thank you again for having me on your podcast.
Well, it's been a pleasure. You take care and have a great rest of the week.
You too. Take care.
Bye-bye.
Bye.
Wow, what a fascinating discussion. I mean, I was really excited to learn all about vaults, and I'm sure you were too. And you can go to our website at natureandsciencepodcast.com to learn a lot more about vaults. And you can also check out Dr. Rome's YouTube channel. It's called The Vault Guy, where he talks about this a lot. You'll learn a lot about cell biology as well. This is just a fascinating discussion, and stay tuned, and we'll be back and talk about this some more and hopefully let you know exactly what vaults do. So, as always, stay safe, share happiness, and remember, never stop questioning. Curiosity has its own reason for existing.